दोस्तों, उम्मीद करें कि आप सब लोग खैर आफियत से होंगे, ठीक-ठाक होंगे। आज आप लोगों के साथ कई दिनों के बाद मुलाकात हो रही है और आज हम लोगों ने एक ख़ास मेहमान को बुलाया हुआ है, जो किसी तारुफ़ के मोहताज नहीं है। हाँ जी, हाँ दोस्तों, आज हम लोगों ने डॉक्टर आशिक़ खासरू साहब को बुलाया हुआ है, जिन्होंने पीएचडी आईआईसी बैंगलोर से कंप्लीट किया था। उसके बाद वो यूनिवर्सिटी ऑफ टेक्सास ऑस्टिन में मूव हो गए, जहाँ उन्होंने डॉक्टर जयराम के साथ पहला पोस्टडॉक किया था। वहाँ उन्होंने साइट स्पेसिफिक डीएनए रिकॉम्बिनेशन पे काम किया था। फिर उन्होंने दूसरा पोस्टडॉक भी उसी यूनिवर्सिटी में ले लिया डॉक्टर मैकाटे के साथ, जहाँ पे उन्होंने इवोल्यूशनरी कन्वर्जेंस को अच्छे से स्टडी किया था और ह्यूमनाइजेशन ऑफ यीस्ट के बारे में जो उन्होंने कंसेप्ट दिया था, जो साइंस पेपर में आया था 2015 में, एक अच्छी ब्रेक थ्रू थी उस टाइम की। तो वहाँ से दूसरा पोस्टडॉक कंप्लीट करने के बाद उन्होंने मूव ऑन किया और फर्दर ऑन वो यूनिवर्सिटी ऑफ कनकॉर्डिया मॉन्ट्रियल कनाडा में इस वक्त फैकल्टी है, जहाँ पे वो टायर 2 रिसर्च चेयर भी है और उनके अंडर 10 स्टूडेंट काम कर रहे हैं इस टाइम, जिनमें कई पोस्टडॉक है, कोई पीएचडीज है, रिसर्च ट्रेनीज हैं। डॉक्टर आशिक़ हम देखें तो उन्होंने साइंस, नेचर बायोटेक्नोलॉजी, ई लाइफ, प्लास जेनेटिक्स ऐसे बड़े सारे नामी जर्नल्स में पब्लिश किया हुआ है और उनका लिस्ट बहुत लंबा चौड़ा जा रहा है। आप लोगों का फर्दर टाइम लिए बिना ही हम डॉक्टर आशिक़ से ही सुनते हैं कि उनकी कहानी क्या थी और उनकी जर्नी कैसी रही है। तो हम वेलकम करना चाहेंगे डॉक्टर आशिक़ का। अस्सलाम वालेकुम डॉक्टर साहब, क्या हाल है आपके?
वालेकुम वालेकुम सलाम जाहिद भाई, कैसे हैं? बिल्कुल ठीक।
अल्हम्दुलिल्लाह। तो चूँकि
थैंक यू फॉर इनवाइटिंग मी।
मैं नहीं मैं आपकी जो अचीवमेंट्स है, चूँकि आप हमारे कश्मीर के साइंटिफिक कम्युनिटी में से एक मतलब ऐसे एक बंदे हैं जिनको किसी तारुफ की जरूरत तो है नहीं, लेकिन चूंकि हम लोग जो आपको पता है साइंस आउटरीच में ये जो है मीडिया की आउटरीच बहुत कम रखते हैं यूजुअली, काम के साथ बिजी होते हैं। फिर भी जो लोग आपको नहीं जानते हैं, आप थोड़ा सा अपनी जर्नी के बारे में उनको बता दीजिए। डॉक्टर आशिक़ आते कहाँ से, उन्होंने अपनी जिंदगी की शुरुआत कैसे की थी और चलते चलते आप एक पीआई कैसे बने?
ऑलराइट, थैंक यू फॉर दिस काइंड इनविटेशन। जाहिद भाई, मैं आया हूँ, मैं 100% कश्मीरी हूँ, ओके। मैं आया हूँ सोपोर से। सोपोर, आई वाज बोर्न इन सोपोर। मेरी स्टडीज भी सोपोर में ही हुई हैं until at least 12th, even college studies were in Sopor. So essentially my bachelor's तक मैं सोपोर में ही रहा, बाहर निकला ही नहीं मैं सोपोर से। और बैचलर के बाद मास्टर्स मैंने किया at MSc Biochemistry at Kashmir University. That was in around 1998-99. So biochemistry में हमारा मास्टर्स हुआ. That time Professor Indrabi, who eventually became the vice chancellor of the Kashmir University, he was a faculty there, and he was a big.
मैं यहाँ पे कहूँगा कि लास्ट टाइम एक्चुअली जब मैंने सिमिर भाई का इंटरव्यू किया था, तो उन्होंने भी बहुत मेंशन किया था इंदिरा बिसर का, कि जब वो
Yes,
आए थे
yes, he
बाहर
was
से।
a huge motivation for many students at that time in biochemistry. So masters वहाँ खत्म हुआ, तो मास्टर जब खत्म हुआ तो उसके बाद we tried for doing further research because of interest in research. और applications हुई JNU, जवाहरलाल नेहरू यूनिवर्सिटी में, NCBS, IISc और बाकी बहुत सारी जगहों पे. Eventually got bunch of interviews. One of the interviews was at IISc Bangalore, which is also called Tata Institute, Indian Institute of Science Bangalore. There, eventually I settled as a PhD student or a graduate student. Continued my PhD for almost 6 years there, and eventually stayed for one more year to finish my paper. So by 2007, I was done, and then moved to Austin, Texas, for my postdoc. The first postdoc lasted for a couple of years, and that was on site-specific DNA recombination. And then the second postdoc lasted for almost 6.5, 7 years. That was on more on evolutionary aspect of studies, how genes are conserved from one species to another. And once I finished that postdoc, वो जब खत्म हुआ तो उसके बाद मैंने application शुरू की for professor position, assistant professor position, and that happened multiple different applications and different, depending on whether I got an offer or not, eventually decided to move to Canada. Right now, that's where I am, calling to
मुझे
you.
यहाँ पे मतलब एक क्वेश्चन जो जहन में आता है कि आपका सीवी देख के हम लोग जब देखते हैं कि आपने वापस बैक होम क्यों नहीं किया? आपको क्या लगा कि वहाँ पे रिसर्च फैसिलिटीज कम है, या जो रिसर्च बंदा एक लेवल पे करना चाहता है वो वहाँ कर नहीं सकता है? क्योंकि आपका सीवी जो है वन ऑफ द बेस्ट फ्रॉम कश्मीर एट लीस्ट.
ऐसा कुछ नहीं है. That was one of the options. In fact, I did apply in India, not necessarily in Kashmir. At
I
least,
heard perhaps about CDFD.
yes, I did. I was offered at CDFT as a faculty position. I kind of gave an interview at NCBI. I tried at Tata Institute of Fundamental Research, Bombay. There also I interviewed, and CDFT offered, and it was a serious offer. I mean, at one point there was a discussion at our home that should I join or not, weighing other options, considering the future of our kid, uh, family issue things, and other things. Eventually, at that time I didn't have any other offer. So imagine an offer is given and I have nothing else to rely on. I had to say no because of other issues, right? So this is what I kind of wanted to convey to students. There are so many things which will happen to you during your course of career development and all. Many times you'll have to take very serious decisions.
So you mean like you are saying to the audience, especially the students who are listening to you here, like, don't give up.
No,
Believe
no,
in
no,
yourself.
no. And many times, you might get a offer or something, which might seem like the best choice at that time, but you should really consider the long-term interests that you should have. Those are the ones which really should drive your decisions. So basically, आपका जो question था कि मैंने वापस क्यों नहीं गया? I definitely had choices, and also wanted to go back. But eventually, for a long-term future development of my career, it was better that I stayed in North America, or generally in America or Canada. So that seemed to be a better option.
No, we can मतलब see like आप इस वक्त one of the best जो कम से कम yeast का हम देखे सेंटर में आप इस वक्त लीड भी कर रहे हैं. तो जहां तक मैं आपका रिसर्च देख रहा हूँ, एक इंटरेस्टिंग फील्ड है, बट अनएक्सप्लोर्ड लग रहा है. जैसे कि एक कॉमन सवाल जो आपके रिसर्च पे देख के याद आता है कि जैसे आप लोग इवोल्यूशनरी रिजेम्बलेंस देखने की कोशिश कर रहे हैं, चाहे यीस्ट और ह्यूमन के बीच में, कि कितना कंजर्वेशन रही है या कितना नहीं रही है. एक इंटरेस्टिंग फैक्ट मैं आपके पेपर से भी पढ़ रहा था कि जहां पे आपने ये मेंशन किया था कि जरूरी नहीं है कि जो होमोलॉगी सीक्वेंस की मतलब जो सिमिलरिटीज है उसी बेस पे हम लोग इसको रिप्लेस करें या ना करें. तो कहाँ तक आपको लगता है कि ये सिस्टम रिप्लेस कर पाएगा जो ऑलरेडी एस्टैब्लिश्ड सिस्टम्स है? जैसे कि अगर हम देखें कि इतने सारे सिस्टम्स ऑलरेडी
Yeah,
इन डिजीज को स्टडी करने के लिए ऑलरेडी
excellent, excellent.
पॉसिबली है.
That is a really good question. But before I delve into that question and answer you, I want to basically give a little bit of a background on this.
श्योर श्योर श्योर
That
श्योर.
might help people understand it better. So basically, the work that our lab is interested in right now is that, first of all, we want to know when you look at divergence of species, essentially like us or monkeys or chimpanzees or Drosophila, flies, mice, and so on and so forth. Yeast is one of the species that has diverged a long, long time ago from the common ancestor. And the common ancestor is the one which was probably between yeast and human, lived between almost 1 billion years ago. So you're looking at a very huge evolutionary distance here. So it's an irony that you, me, and many other Kashmiris came from a place where we never learned about evolution in entirety of our career. So when I learned about evolution, that was only in IISc Bangalore when I took a class there, and got introduced this lovely field of biology about how evolution happens. What is the meaning of evolution? So the concept is that long time ago, we had a common ancestor, which is us and many other species, including this yeast, which we use in making bread. So that common ancestor had a set of genes, okay? Set of genes that it used to live. And those genes were passed on to next generation, so on and so forth. Eventually, after a billion years, yeast is what we have right now, and we are humans. But we share several thousand genes because of this similarity, okay? Not just 1 or 10. Yeast has only 6,000 genes. So imagine a simple eukaryote with 6,000 genes, and almost 1/3 plus genes have shared orthology or similarity between humans and yeast. So that's remarkable. So even if there's such huge 1 billion years divergence, you still have shared genes. So that people know for a long time. It's nothing new. So this is not anything earth-shattering. We know that. Biologists knew that for a long time. But what we don't know is when we say something is a shared gene, that doesn't mean it does the same exact function, okay? So our work that we did, that was what you mentioned, the science paper, was to actually test that. What we call as shared genes, are they really doing exact same function? That was done by the common ancestor a long time ago. So the test was that if you take the human gene, which we call as a shared gene, and then put it in the yeast and remove the copy of the yeast gene at the same time. So essentially, yeast is living because of that gene. You remove that gene, it dies. And now you put a human copy of that gene, and you want to say, can you live with the human gene? If it lives with the human gene, that means that yeast gene that you deleted was doing exact same function as this human gene is doing. Therefore, it's covering for that function. It is literally saving yeast from dying. That was a simple test. What we did with that test was we tested not for one gene, but nearly now almost 1,000 genes.
Yeah, that I mean, it is like 414 genes in
Yeah,
that
that
paper.
was in science paper. Eventually, we went on and finished more sets of genes. We have by now targeted, literally tried it for nearly 1,000 genes. And remarkably, what we found was that over half of them, tested genes that we tried, actually,
Work.
save yeast, work exactly the way yeast gene was supposed to work. So that is the beauty of this work. Not only do we know now that shared genes exist between one species and another, but also they can cover for the function. They exactly do the same function. Turns out these are very important genes. They are the basic building blocks of our cells. They don't diverge much. Therefore, they retain these interesting, important functions. Now comes the question that you asked. You're asking me, why are you doing this in yeast, right? What is the point of this? There are so many other awesome models, more close to humans than yeast is. The beauty of yeast is that it's a simple cell. You can do lots of genetic manipulations with it. It's easy to grow in the lab. You don't really need a big NIH grant to do this research. So you can easily manipulate these human genes. You can mutate them and do whatever you want to do with this easily in this simple cell.
Like you don't call it ethical
Right. You
committees
don't
as
have
well.
ethical concerns also, exactly. So the idea is that if you now have this human gene covering for the human, yeast gene function, you mutate it. This mutation could be an important mutation, let's say a cancer causing mutation, or a disease in humans, which results in some kind of Mendelian disease or some other disease. We can actually study this mutation. What exactly does it do? You can mutate it any other residue or any other amino acid, mutation. You can do this mutation for not just one amino acid, but almost entire protein. You can scan this protein entirely, study this in this simpler cell. So that's the power of yeast, okay, which you cannot do in humans. You don't
I have a
have any.
question here, like how much homology did you usually find when you kind of replace gene which had exact function in the yeast?
So homology, as I said, if you look at the number of genes that we share between humans and yeast, you're looking at almost 4,000 human genes, which have shared orthology or similarity in the yeast with the yeast gene. So we know that's a huge number, and only tested with 1,000 so far. Okay. So what are these genes? 4,000 genes. Turns out they are important genes. As I said, they are the basic building blocks of our cell. And if you mutate a basic important gene, no matter which species you're talking about, that leads to disease, right? And in the case of humans, almost every gene that we call a shared gene has a disease-associated phenotype. That if it gets
लेकिन
mutated,
मुझे और एक चीज याद आ रही है यहाँ पे कि like if we are tomorrow kind of a I guess perhaps reverse has not been studied yet. Like, suppose if somebody is dying and we are putting that particular yeast in there, so
exactly.
how that will behave in
Yeah,
the
that's a
vicinity?
fantastic question. Now you're talking about serious questions here. So you're asking me the reciprocal test.
Of
If the
course.
human gene works in the yeast, would the yeast gene also work in the human context? And that has not been done so much. In fact, if you look at the literature, one or two studies, couple of them, and one of them worked. So you can take a human yeast gene and put it in the human, and it actually does exact same function. Systematically, it has not been tested yet. Something that I'm interested in personally, in fact, to be frank, there are a bunch of projects going on with the collaborators, which is testing exactly this, whether the reciprocal thing can happen. Can you take a yeast gene and put it in humans? What is the value of that research? I don't know. Maybe if you look at the fundamental aspect of this, that can you put any other species genes, no matter what it is, if it's a shared gene, you can put and cover the human gene function. So that might be fundamentally important for understanding the basic concept of evolutionary conservation. But what application would it have? One aspect of application that comes to mind is that these are shared genes. That doesn't mean they're exactly same. They're not identical genes. They are similar. In other words, if you
I
look
was
at this,
thinking about like what will be the fate of their post-translation modifications.
right?
What will the fate of other important cofactors which will be playing part because the
Right.
environment especially environment will be totally different.
Yes. So to be frank, when we took the human genes in the context of yeast, yeast doesn't have the same exact post-translational modification that humans have. Yet many of these genes still work. They didn't need those post-translational modifications to function in the context of, at least in the yeast context. But the same thing may not be true. We might be surprised when we try to put the other species genes which have diverged significantly from humans in the context of humans. They may not actually work. You might need these post-translation modifications. So that's a question which is not yet answered, and something that people should, at least we are interested in, and somebody should do that.
Yeah, that's actually that's what will come. Being
Yeah.
a scientist, that will come. First thing will come to the mind that what will be actual applicability when it comes to the humans, if reciprocal thing does work or not.
Yes.
But
Yes.
now, interesting question regarding your research here will be like when you are kind of studying it, like what are the mutations, particular mutations, all those things. Do you think whatever you have studied in the yeast there could have similar effect back in human as well?
That's a fantastic question. There you go. You give an impression you're a very smart guy. So kudos to you. So what Javed is asking for the people who are not yet understanding this question is that now that I take a mutant gene from humans and put it in yeast and see the effect in yeast, how does it correlate in humans? Well, that doesn't mean anything. Yeast, it shows some effect. It is not necessarily going to show exact same phenotype in the context of human cell. That's a genuine question. That's a genuine, what you call as a drawback, if you can say that. But I will give you a counter argument with that, or counter effect on that. So we don't have any other choice, number one. For example, by now also, you can get your genome sequenced tomorrow for $99. And that's what almost many people are doing. In fact, right now, we have thousands of human genomes already sequenced. They're already put in NCBI, the database, the place called Variant Viewer. So what it turns out, what we have is, although we are the same human species, our genes differ. They don't have exact same sequence. So we are putting these gene sequences everywhere in the genome databases. We find lots of different changes in our genes. Nobody knows what these changes do to these genes. Are they normal polymorphisms? Are they serious mutations? We have no idea. And when you look at that database, it is in thousands of variants. Like scale is going every day more and more. So how do we study the effect of these variants or these changes in our genome sequences easily? One choice you say you could do it in humans. So mutate every one of them and
Because
test the function.
why this came to my mind, because during my PhD I was working on SDAX,
Yeah,
and when we particularly look into the SDAX genes, when you replace quite a few amino acids, activity drops by thousand folds
yeah,
at certain places.
yeah,
That's what will if it will come out like we have been working in a yeast and we have been working on the particular mutation or a modification, and we'll come
yeah,
tomorrow with the concept that let's see this gene is having a mutation A, and we have studied it in yeast, and tomorrow when we will be targeting that mutation A in the human for that particular gene, and it comes out that it was A plus B, it was not A only.
yeah, yeah, yeah. So
So
these are called epistatic effects. So what I wanted to finish that question. So essentially, we have a huge deluge of data already there. We don't have a choice but to test them out. You can try in humans, but it's costly. Uh, it is difficult to do so much at that scale in humans. So we have no choice but to use simpler cells. So one such system is yeast. We can actually do lots of deep scanning mutagenesis, what they call. Every amino acid can be changed to every other amino acid. So essentially every mutation, essentially 20 different mutants can be created for every residue. You can do those things in yeast. It's really easy. So that allows feasibility. So we can try it in a simpler cell and see. That's one question. Second question is, how serious are we? Are we really getting information by trying these genes in yeast? The answer is yes. So the answer is based on two aspects of the research. One is that the only other choice that we have at this point is to look at structural data. So basically predict if the gene has this structure and this mutation happens, what is the impact of that mutation on the structure of the gene? And if the structure changes, the likelihood is that it might affect the function. So if you look at that prediction, and people have done that, and compare it to the data which we know associated with disease, and compare that with the data in the context of the yeast cell when we try to humanize the yeast gene, it turns out yeast does far better, like far better in terms of predicting whether the gene has an impact on the function, considering if you look at the structure analysis. So clearly yeast is winning here. Your other choices do not really work that well. So this is what my thinking is, that one is feasibility, we can do these things in the context of these. Second is that we are actually doing close to good in predicting whether the gene has some serious function. And third one would be the era of systems genetics, what we call synthetic biology. Yeast is a absolutely fantastic model to not just put one gene, but actually put a system of genes in the cell. So your problem could be, if you are a critic for this research, you will say, look, you put just one gene, every other gene is still human, yeast in the context of yeast. So it doesn't really make sense. Genes don't work alone. So the answer to that is that we are actually putting not just one gene, but the entire system. For example, in the lab right now, you might know one of a Kashmiri student, Mudabbir, is working with me. He is not just putting one gene from one system, he is actually engineering the entire biological process in the context of yeast cell. So that might be more relevant. So when you have the entire, what we call as local interaction, local genetic interactions, exactly same as you expect.
actually, you at this point gave me answer to that I was going to ask. Actually, what I was looking at abstract.
Yeah, so you should be able to
Yeah.
answer those questions much more seriously. Essentially, we're not sitting back and saying, okay, this is what we did and this is going to be perfect from the
Should we
context of
assume
human.
that this systematic biology is still at its infancy stages?
Not in US, in other places. The US actually has picked up significantly. Maybe in India it is in infancy, but definitely several labs are doing systems biology and synthetic biology. Almost everyone does. They just don't know they're doing it. You probably do synthetic biology and just don't know that you're doing it.
Okay, that's an important observation actually. Yeah, that's actually. It looks like being a student of a science. I would love to like listen to this conversation on and
Yeah,
on,
yeah, yeah.
because it looks like we are getting this beautiful lecture that how it goes on since. we had to do other part in the video as well. Like I know that the whole student community who will be watching back home will be waiting for something else. तो अगर हम अब इस चीज की ओर आ जाए कि जैसे कि क्योंकि आप इस वक्त नॉर्थ अमेरिका में काम कर रहे हैं, आपने इंडिया का सिस्टम भी देखा हुआ है, जो जब लोग पीएचडी के लिए जाते थे वहां पे स्पेशली, या जब आप कश्मीर यूनिवर्सिटी से निकले तो आप कह रहे हैं कि आपके लिए एक इंस्पिरेशन थी कि डॉक्टर इंदिरा भी उस टाइम आए हुए थे, जिन्होंने काफी लोगों को पुश किया कि नहीं भैया आप लोग चले जाए, बाहर जाए, आप एक्सप्लोर करें। तो वो एक्सप्लोर करते करते आप बाहर आए और फिर इवेंचुअली इस वक्त एक सक्सेसफुल साइंटिस्ट है आप, जो एक सीरियस मतलब एक ऐसी प्रॉब्लम को सॉल्व करने की कोशिश कर रहे हैं जिससे बिल्कुल बायोलॉजी का एक पूरा पैराडिग्म जो है बदल जाएगा, अगर हम इसको पूरी तरीके से सक्सेसफुली अचीव कर गए। तो यहाँ पे जब स्टूडेंट एक जो रहता है उसको किस स्टेज पे आपको लगता है कि सीरियस हो जाना चाहिए कि मुझे साइंस के लिए जाना चाहिए कि नहीं? क्योंकि जहां तक मुझे पता है अभी हम वहां कहते हैं कि हमें कुछ नहीं है फिलहाल, चलो मास्टर्स कर लेते हैं, जॉब नहीं है उधर से पीएचडी की तो चलो पीएचडी कर लेते हैं। मैंने समीर भाई से लास्ट टाइम जब पूछा था तो उन्होंने बोला था कि मैं बीएससी लेवल पे ही जाऊंगा कि और उन्होंने जो है एक पर्टिकुलर जो टैग उन्होंने मारा था, उन्होंने कहा था कि एमएससी के बाद आप बिल्कुल घर बैठे 3 महीने 4 महीने, जो आपने भी कहा कही थी कि मतलब अपॉर्चुनिटीज आती रहती है, लेकिन आपके लिए पीएचडी लैब आपकी डिफाइनिंग लैब हो जाएगी कि आप कहाँ जा रहे हो, किसके साथ कर रहे हो, ताकि ये आपकी एक लाइन बन रही है, बाद में आप भले ही चेंज हो जाओगे लेकिन आपको टाइम लगेगा। तो
Yeah,
आप उस मामले में क्या कहना चाहोगे यहाँ पे
yeah,
स्टूडेंट्स से?
yeah. So I think I don't.
तो दोस्तों, ये इंटरेस्टिंग टॉक जो डॉक्टर आशिक के साथ जारी है, इंशाल्लाह हम पार्ट 2 में देखेंगे कि स्टूडेंट्स के लिए इनकी खास क्या एडवाइस है, उनको क्या करना चाहिए और कैसे कैसे अप्रोच ज़िंदगी में उनको अपनाने चाहिए। तब तक के लिए आप सब लोग अपना बहुत सारा ख्याल रखेंगे। इंशाल्लाह फिर मिलेंगे पार्ट 2 को लेके। अस्सलाम वालेकुम।